and okay, so we just started recording. So, hi, so my name's Reef. I'm a fifth year medic. So I'm just going to get started now because it otherwise it's going to be a little bit too late, but hopefully more people will trickle in as we get going. Um, so I was given the very broad title of genetics, which is about a third of what you do in first year. So I worked quite hard to try and condense this all into an hour length lecture. Um, now, I'm mostly going to focus on molecular genetics because I imagine that you did this most long ago, whereas the sort of medical genetics part of it is more recent. Um, so briefly, I'm going to cover sort of the DNA and chromosome structure, DNA replication, and then our main processes in gene expression. And then I'm going to also touch on the different methods we have for gene and gene expression. So this is everything that's sort of covered in 3.1 in your syllabus. Now, we've got a lot of slides and a lot of content to get through. I've also got some questions dotted throughout. I would say don't worry too much about taking notes. So everything that I will be saying is either going to be on the slides or in the speaker notes, and this is all going to be sent around. In terms of how I focus this, I've really tried to keep it to your part A's, and obviously all of this will be helpful for your part B's, but I haven't put in any experimental evidence. There's odd clinical conditions that I've mentioned, but that's not really the focus of this. All right, so first of all, defining a gene. So these are our inherited units of information. So they either specify phenotype at a really gross level. They might code for things at molecular level. So our various RNAs and proteins, right? And as we know, the vast majority of the genome isn't made up of genes. Okay. You don't need to know the prokaryotic gene structure, but just include it for completeness. And this sort of structure for the eukaryotic gene will again come back to in a little bit. Thinking a bit more about DNA structure. So DNA and RNA are nucleic acids consisting of a long polymer of nucleotides. Now, each nucleotide has our nitrogenous bases, of which there's two types. So we have our purines, which are adenine and guanine, and we have our pyrimidines, which are cytosine and thymine in DNA and uracil in RNA. Now, I was getting confused. I would always think small word, big molecule. So adenine and guanine. So purine, smaller word. So adenine and guanine are the bigger ones. Now, apart from the nitrogenous base, we also have our pentose sugar, which is deoxyribose in DNA and ribose in RNA. And then we also have our phosphate group. Now, if we could think about what a nucleoside is, you can see right at the top that the only difference is that a nucleoside doesn't have a phosphate. So just if you see the two times used, just so you can know the difference. Now, DNA exhibits polarity. So we always think about it from 5' to 3'. And this refers to sort of the free carbon atom of the sugar, which is free at the end of the chain. So the 5' end has a phosphate group on the C5 of the deoxyribose, and the 3' end has a hydroxyl group of the deoxyribose. And we think about our DNA strands running anti-parallel, so they run in opposite directions. And the last thing is that we have our base pairing rules. So adenine always pairs with thymine or uracil in RNA, and that's with 2 hydrogen bonds, and cytosine pairs with guanine with 3 hydrogen bonds. And this is our complementary base pairing, which underlies all of this. So now thinking about sort of the more macro structure. So our nucleosome is our basic unit of chromatin. So this is our polynucleotides with the histone octamer. So the histone is made up of the 4 subunits, H2A, H2B, H3, and H4. And this forms our octamer. And so DNA is associated with histone proteins, and the histone arrangement determines DNA replication. And so when we think of nucleosomes, that's our basic unit of chromatin. And so that then goes up into our macro structure as well. So within chromatin, we have our euchromatin, which is our loose active during transcription, and our heterochromatin, which is tight and inactive. And within heterochromatin, we have our 2 types as well. We have facultative and constitutive. So facultative can become transcriptionally active. So depending on epigenetic changes, that can become euchromatin. But our constitutive heterochromatin is repetitive and forms structures such as our telomeres and centromeres. So now thinking about how mutations can occur and come about. So first of all, a mutation is just a change in the nucleotide sequence, and it can range all the way from sort of single nucleotide changes to whole gene duplications or deletions, right? And we generally think about how this can occur spontaneously or whether it's induced by mutagens. So first of all, our spontaneous mutations. Our first one is deamination. So that is just the loss of the amine group in our nitrogenous base. And this occurs via a hydrolysis reaction in the aqueous environment of the nucleoplasm. So for example, we see that if you look at the top, you can see the deamination of cytosine to uracil. And uracil is, of course, not normally present in DNA. And so this mutation can propagate unless it's corrected prior to DNA replication. And that's because, as we'll talk about later on, that both strands act as templates in our semi-conservative DNA replication. And so this is going to be propagated in one of the DNA strands. And so because of that, as adenine is complementary to uracil in RNA, that's going to now have an adenine instead of a guanine. And so we have a literally like single nucleotide replacement of CG base pair with a UA base pair. Alternatively, we also often have cytosine, which is methylated as part of an epigenetic change, and their deamination completely changes it to thymine. Now, this won't be recognized as abnormal because thymine is just a normal component of our DNA. And so this won't be recognized by our DNA repair enzymes, whereas uracil could be recognized by our DNA repair enzymes. And this is part of our sort of high fidelity. Another mutation that we get is our depurination. And so that's when we have a purine base that is just lost from the nucleotide by a hydrolysis reaction. And so the single unpaired base is deleted or substituted from the DNA, and this can lead to a frame shift if it's just completely deleted. And so that changes the reading frame, and so everything downstream of that change will now have a different codon, and so a different amino acid, and so on and so forth. The last thing I'm going to talk about on this slide is our ultraviolet radiation. And so what this can do is the UV radiation can cause thymine dimerization. And so that's a covalent cross-linkage between our adjacent thymine residues on the same polynucleotide strand. So you can see that, hold on, that here you have this covalent linkage. And so this affects the DNA structure. If you can imagine it, it might cause some kinking in the phosphodiester backbone. And so this inhibits polymerases. And what that means is that it halts replications. We do have some repair mechanisms, but they're not always effective. And if you have an inherited condition where these repair mechanisms are faulty, then it really increases your likelihood to get mutations from UV light. So one of these conditions is called xeroderma pigmentosum. And again, don't worry, this is all in the speaker's notes. But essentially that involves mutations in the genes coding for the proteins that repair UV light damage. And so in people with xeroderma pigmentosa, exposure to UV light triggers like a high frequency of mutations in skin cells, which in turn results in a really high occurrence of skin cancer. And so as a result, individuals with this condition are really unable to go outdoors during daylight hours. Now, let's think about substitution mutations specifically. So we have three broad types, which are our silent mutations, our missense, and our nonsense mutations. So within the DNA code, each three nitrogenous bases codes for an amino acid. Okay. Now, in a silent mutation, the change in the nucleotide does not change the amino acid. It's translated because the genetic code is degenerate, right? So with our four bases and our codon having three, being made of three nitrogenous bases, that gives us four possible triplet codons. But because we only have 20 amino acids, several codons may code for an individual amino acid. And so in a silent mutation, it's just another codon for the same amino acid. A missense mutation alters a codon, and so that leads to an altered amino acid. Now, this may or may not affect protein function depending on the chemical properties of the new amino acid. So if it has similar chemical properties, such as possessing a positive charge, for example, arginine for lysine, this may have very little effect on protein structure. So this might be a conservative mutation. If they're not similar, then bond formation and overall protein structure can be highly disrupted. Many of the amino acids are just scaffolding within the protein. And so if the catalytic residues of the protein are in the correct space, substitution will have very little effect. But if they are in the catalytic activity of the protein, such as being in the active site of an enzyme, then protein function will be altered. And lastly, a nonsense mutation introduces a premature stop codon. So those are our UAA, UAG, and UGA in the mRNA. Now, this prematurely stops translation and so it produces a truncated polypeptide chain. And this can be a non-functional or dysfunctional protein. These aren't usually formed because we have a safeguarding mechanism called nonsense mediated mRNA decay. And so this decays mRNAs that contain nonsense mutations before they can be translated. So it's one of our surveillance mechanisms. And what this relies on is something called exon junction complexes. And these are proteins that attach to mRNA at exon-exon junctions following splicing. And so the presence of an EJC downstream of a stop codon identifies that the mRNA is faulty and so induces its degradation. Okay, the next type of mutation we're just going to talk about briefly is indels. So that's insertion or deletion. And so this can either be a multiple of 3 or not a multiple of 3. And so that if it's a multiple of 3, then everything remains in frame. And so you just have the addition or deletion of a number of amino acids. And so severity depends on the protein and the amount of amino acids that are inserted. So for example, in cystic fibrosis, you have a deletion of just 3 nucleotides in the CFTR gene, but that results in the loss of phenylalanine, the 508th position in the chloride channel. And so this leads to abnormal folding, subsequent degradation of the protein before it can reach the plasma membrane. This means chloride doesn't move out to the cell and so prevents the loss of water from cells by osmosis. And that's what causes sort of thick, dry mucus that can build up in organ passages in cystic fibrosis. Now, symptoms that come with this is abnormal pancreatic function, frequent chest infections, and coughing. Now, if we think that it is a non-multiple of 3, then what we get is a frame shift. And this can be very serious on the protein structure. So this shifts the reading frame and alters every downstream triplet and thus amino acid. And often what occurs is that there's introduction of a premature stop codon. So one of these conditions where this occurs is Duchenne's muscular dystrophy. So this is an X-linked recessive disorder that mainly affects young boys. It's commonly caused by a deletion mutation in the gene that encodes for dystrophin. But this deletion causes a frame shift, which introduces a stop codon, so our nonsense mutation. This causes premature termination and degradation of the dystrophin protein. As a result, this impairs transmission of force from muscle to bone in patients with this condition. Excuse me. And so symptoms include muscle weakness and reduced motor ability. So there are potential treatments that are being researched, including exon skipping, in which like antisense oligonucleotides are used to try to restore the reading frame. Now, when we think overall about the various mutations that occur, we can think about whether there's a gain of function or loss of function, or there's no change in function. So gain of function can lead to excess protein production by increasing normal gene function, so a hypermorph, or a novel protein production, a neomorph. These mutant alleles will be expressed even in the presence of a wild-type allele, so they are dominant mutations. Loss of function mutations can lead to reduced or no protein production, or production of a non-functional protein. And so these mutations are typically recessive, as the presence of a wild-type allele can usually produce a sufficient amount of protein to retain the normal phenotype. An exception for this is dominant negative mutations, where the abnormal protein product interferes with the normal protein product and inhibits its function, i.e. by sequestering part of the protein. Okay, so just thinking about DNA structure and mutations that we've talked about so far, there's some questions here. Now, I don't know if people want me to have a read of it now, put an answer in the chat or respond, or otherwise I can just read the answers out. I'm afraid I have a lot of things to get through in this presentation, so I won't have time to exactly explain each of the answers, but after, if there's any confusion, I'm very happy to be emailed. Okay, so does anyone want to contribute for the first answer? So the mutation type where all amino acid sequence downstream of the mutation resulted? Yeah, good. So that's frame shift. Absolutely. And what about the rest of the questions? If not, I'm quite happy to also just read the answers. Sure. So, hold on. DC, F, and B. Good. Yeah, that's exactly right. So first is frame shift. The second is a silent mutation. Third is a nonsense mutation. The fourth is X-linked, and the fifth is a missense mutation. All right. So now let's talk about DNA replication. So this occurs during the S phase of the cell cycle. It's a process by which a single DNA molecule is copied, producing two identical DNA molecules. And so in eukaryotic cells, DNA replication generates two identical copies of a chromosome that are each inherited by one of the two daughter cells produced by the mitotic cell division. And so this means that the daughter cells are genetically identical to the parent cell. Now, the key thing is, as mentioned before, that this is semi-conservative. So one DNA strand from the original parent molecule and one newly synthesized DNA strand goes to each daughter cell. And as you'll hear me say about 100 times in this presentation, this process relies on complementary base pairing. So thinking about it, there is primarily three steps: initiation, elongation, and termination. Let's talk about initiation. So in eukaryotic cells, replication is initiated at origins of replications, or ORI, which are specific sequences at specific sites from which replication occurs in both directions along the DNA molecule. Now, each chromosome will have multiple ORIs, which increases synthesis rate and decreases replication time, which is necessary because the DNA molecule is so long. And so this pre-replication complex is assembled at ORIs during G1 phase of the cell cycle, and ORIs are activated throughout the S phase, and this is very tightly regulated. The origins of replication recruit DNA helicases, which open the DNA double helix by breaking our hydrogen bonds between the complementary base pairs. And so that produces local separation of the polynucleotide strands. So single-strand DNA binding proteins, for example, replication protein A, associate with the exposed strands, and that prevents them from rehybridizing. So then once we have this, we have the structure of a replication fork. And so this is the junction between the double-stranded DNA and the separated strands. Now, as the DNA is unwound, there's tension in the replication fork. And so DNA gyrase, which is a type of topoisomerase enzyme, relieves that tension and stops the unwound single DNA strands from getting tangled by a breaking and rejoining mechanism. So now thinking about how is this new DNA strand actually synthesized? So there are two enzymes here that we're thinking about. We're thinking about DNA primase and DNA polymerase. So DNA polymerase catalyzes the synthesis of new complementary polynucleotide polynucleotide strands using the pre-existing exposed strands as templates. So DNA polymerase, as we'll see for other types of polymerases, synthesizes in a 5' to 3' directions. And this catalyzes the formation of a phosphodiester bond between a deoxynucleotide triphosphate and a terminal 3' of a deoxynucleotide monophosphate residue in the new strand, or DNTP and DNMP. But DNA polymerase requires an existing strand to initiate synthesis from. It cannot initiate a new strand of DNA by itself. And this is where DNA primase comes in. And so this is an RNA polymerase that synthesizes short RNA primers about 10 to 20 bases long on the template strands. And this provides a 3' OH for DNA polymerase to extend. And so it's therefore critical for this process. Now, there are a few other proteins that are involved. So there is replication factor C and PCNA, which form the DNA clamp or a sliding clamp. And it's a protein complex that binds DNA polymerase and prevents the enzyme from dissociating from the template DNA strand. It's also involved in increasing processivity and very high affinity. And so without it, we'd have a much slower process and increase of error. So due to the DNA strands being anti-parallel, the opening of a replication fork exposes new regions of template strands that run in opposite directions. And so this is our leading strands and our lagging strands. So the new leading strand is synthesized continuously in the 5' to 3' direction. And it's the same direction as the movement of the replication fork, if you can imagine here moving into there. And so as DNA polymerase can directly extend the strand in this direction, it also only requires 1 primer. And so the lagging strand is, if we imagine moving this way, because DNA polymerase can only synthesize in a 5' to 3' direction, is having to resynthesize overall in a 3' to 5' direction, but in Okazaki fragments that are each synthesized 5' to 3'. And so these lagging strands are synthesized discontinuously, but towards a replication fork. And so this requires multiple primers. And so thinking about our DNA polymerases, we have two slightly different isoforms that synthesize the lagging strand and the leading strand. So Pol delta conducts the majority of discontinuous lagging strands Okazaki fragment synthesis, while Pol epsilon accomplishes the majority of continuous leading strand synthesis. And so just talking a little bit more about our Okazaki fragments. So again, this is just in our lagging strand synthesis, and we need our DNA primase to repeatedly synthesize new RNA primers. These Okazaki fragments are about 150 nucleotides long, and so you might imagine that there's going to be several fragments produced during DNA replication. And so as the Okazaki fragment is synthesized towards the 3' end, it eventually displaces the previous RNA primer, forming a sort of overhanging flap. And so this is removed by an enzyme called FEN1. Once that's done, the DNA ligase, another one of our enzymes, catalyzes the formation of a phosphodiester bond between the Okazaki fragments and the growing strand, and joining them together. So that's the majority of DNA replication. But what would happen is that with each, because of the Okazaki fragments, and I'll explain this a little bit more in detail in a second, we would have shortening of our chromosomes with each time, which would cause replicative senescence. And so we have telomeres that help protect the very ends of our chromosomes. And so telomeres are DNA protein complexes consisting of a tandem repeat sequence of DNA and a shelterin protein complex. So the tandem repeat sequence is TTA-GGG. And so telomeres are important for differentiating the ends of chromosomes from DNA double strand breaks to prevent end-to-end fusions. So what happens is that as replication reaches the telomere, the replication fork opens. And so this produces free ends. Now, since lagging strand synthesis occurs in fragments of barely consistent length, there must be a distance of around 150 nucleotides behind the 5' end of the last Okazaki fragment and the RNA primer of the next fragment. But at the very end of the chromosome, there are fewer nucleotides than required, meaning that a primer cannot be laid. And so the last few nucleotides of the lagging strand template cannot be copied. And so this is what I was talking about with the daughter chromosomes would then be shorter by their parent chromosomes by around 100 nucleotides in each telomere. And so with each replicative cycle, these chromosomes become progressively shorter until the cell reaches replicative senescence and dies by apoptosis. And so the way we overcome this is with our telomerase enzyme. And this is a reverse transcriptase enzyme, RNA complex, that lengthens telomeres by extending the end chromosome lagging strand template. And this enables the synthesis of the final Okazaki fragment. So how it works is that the RNA molecule of telomerase acts as a primer for lengthening the telomeres, and it contains numerous repeats of the AAU-CCC that is complementary to the tandem repeat sequence found in telomeres, and so that enables hybridization. Exposed RNA bases hang off the end of the chromosome and then act as a template for the enzyme component of a telomerase to synthesize a new complementary DNA strand at the 3' end of the chromosomal lagging strand template. And so this allows us to restore the original length of the chromosomal DNA. So the last thing to think about is what happens when this goes wrong, and what mechanisms do we have to prevent that? So generally, DNA replication is very high fidelity, and the likelihood of a non-complementary nucleotide being incorporated is about 1 in 10 to the 9. And this occurs partly because DNA polymerase also has a 3' to 5' exonuclease activity, which essentially allows it to proofread and correct mistakes. So what that means is that if an incorrect nucleotide is inserted, base pairing will be disrupted, causing a shift in the DNA polymerase from the synthetic state to the exonuclease state. And so this enables the polymerase to remove the incorrect nucleotide by cleaving it before it can then carry on its synthetic activity. Now, what can also occur is that if there is a repeat sequence, you can have replication slippage. And that's where DNA polymerase slips while repeating these repeat sequences. And so this can lead to the insertion or removal of additional repeat units, which can lengthen or shorten the repeat sequence respectively. And this can also lead to a frame shift if the length of the repeat is not a multiple of 3. And one condition where we see this is so this is an autosomal dominant neurological disorder. And so a mutation in the Huntington gene, which encodes the protein Huntington in the brain, leads to the elongation of the CAG repeat and produces an abnormal Huntington protein. So in unaffected individual, this has less than 36 repeats, but in affected individuals, this gene contains 36 repeats or more. And the number of repeats changes in successive generations. And what you can have is the phenomenon of anticipation, where the age of onset of disease is earlier in subsequent generations, and the phenotype becomes progressively more severe. And so symptoms of this include sort of athetoid choroid movements, impaired communication, and depression. Okay, so how are we doing? We're doing all right. So I think for this, let's see if we can just quickly go through these questions. So first, DNA strands are anti-parallel. This means that which option is correct? If people want to speak up or put in the chat, either is fine. If people prefer for me to just go through the answers, I'm also happy to do that. So I'll tell you what, I think because there's a lot to read here, I'll just I won't go through these questions. And then afterwards with the slides, you'll have it for yourself to go through as well, to test yourself. Okay. So now on to our next major process, which is transcription. So this is our first part of protein synthesis, and we're primarily going to consider the process by which a pre-mRNA molecule is synthesized from a DNA template. So this occurs in the nucleus and is catalyzed by an enzyme RNA polymerase II. And like DNA replication, it has three phases: initiation, elongation, and termination. So your syllabus wants you to know a little bit more about RNA polymerases. So there's a number of similarities and differences with DNA polymerase. So like DNA polymerase, they read DNA in a 5' to 3' direction, but they don't have any proofreading ability to know exonuclease, and they also do not require a primer. And then we have different isoforms of RNA polymerase that just transcribe different RNAs. And then there's different RNA subtypes, which I won't go through right now, but you can have a look through afterwards. So comparing to prokaryotes, eukaryotic RNA polymerases require several additional proteins to associate with promoter sequences. And this is where our transcription begins. So these are our general transcription factors. And so GTFs recognize specific sequences located upstream of the genes to be transcribed, bind to the promoter sequence, and recruit the correct RNA polymerase. And each promoter region is recognized by a specific set of GTFs that enable interaction with the correct RNA polymerase. So transcription begins with assembly of the pre-initiation complex. Now, from what I know, this is actually extension material, so I'm just going to go over it quickly, but you do have more detail in the notes. But essentially, you have binding of the Tata box binding protein and its associated factors, which are known collectively as transcription factor 2D. And this is a promoter sequence, and this causes sequential recruitment of our other transcription factors for RNA polymerase II, which are transcription factor 2A, 2B, and 2F. And the binding of these transcription factors leads to eventual recruitment of RNA pol 2, transcription factor 2E, and 2H. And again, that's all in the notes. Now, once our pre-initiation complex is assembled, another one of the general transcription factors, TF2H, has a CDK7 subunit, and this has kinase activity, which phosphorylates serine residues and the C-terminal domain of RNA pol 2, and this triggers initiation of transcription. Other subunits of TF2H have helicase activity and unwind the DNA. Now, in addition, activators and co-activators are required to ensure correct and regulated gene expression, but these bind to regions outside the core promoter elements and ensure timely and controlled expression. Now, what I would also add is that we'll talk about pre-mRNA processing in a second, but the C-terminal domain of the RNA polymerase is the landing pad for the processing factors, and this is what allows pre-mRNA processing to be a co-transcriptional process. So then we have elongation and termination, and so this largely works quite similarly to DNA replication. So RNA pol 2 selects the correct free ribonucleotide trisophosphates and catalyzes a phosphodiester bond between adjacent ribonucleotides. And it's repeated many times with RNA pol 2 moving away from the promoter region in the 3' to 5' direction and synthesizing in 5' to 3'. Now, normal base pairing rules apply, apart from, of course, adenine binding with uracil instead of thymine. And so rather than in DNA replication where you have multiple origins of replication, in transcription it's completely processive. So a single enzyme transcribes a complete pre-mRNA. And so something else that's worth noting is that because there's a reason why there's no proofreading mechanisms in RNA polymerase, and that's because it's largely not needed. Because RNA isn't inherited like DNA is. And so if there is an error in it, there will just be possibly, again, nonsense mediated decay or degradation of the protein. But another RNA molecule can be produced that will not have an errors and produce an eventual functional protein. So then termination is indicated by stop signals. So these signals come actually from pre-mRNA processing. And so what you have then is destabilization of the newly synthesized RNA and the DNA template. And so this causes dissociation of the RNA from the DNA template and of RNA pol 2 from the RNA. And so the DNA strands then reform their double helix. Now, you also need to know about how gene expression is regulated. So this happens at three main points that your syllabus cares about. So that's the promoter. We're looking at DNA and histone modifications and co-transcriptional modifications. So, as I mentioned before, in addition to the general transcription factors, activators and co-activators are required to ensure correct and regulated gene expression. So these bind to regions outside the core promoter elements, such as to enhancers. And so enhancers increase gene transcription by binding to promoter regions such as the Tata box and increase their basal activity and enhance transcription. Silencers, in contrast, decrease gene transcription. And then insulators, which are unfortunately not on this diagram, regulate gene expression between neighboring genes. And that might be relevant, for example, for quaternary proteins that require multiple polypeptides of different genes. Now, long distance interactions between regulators and promoters, as you might imagine, could be several thousand nucleotides apart or more. So this is mediated by DNA protein and protein-protein interactions. And so how this works is that the DNA between the regulator and promoter loops around a DNA bending protein. And this brings the sequences closer together and allows them to interact. So now thinking about histone and DNA modifications, we know that DNA is compacted into chromatin and that not all DNA is always readily accessible. So thinking about our euchromatin and our facultative heterochromatin, we can move about between them through a number of processes. And so depending on how tightly packed the DNA is, affects transcription because it influences the accessibility of DNA to transcription factors. So one way chromatin conformation is altered is by modification of end-terminal histone tails at the nucleosome. So methylation of the histone tails leads to association of nucleosomes to heterochromatin protein, forming heterochromatin and causing it to wrap more tightly. And so this causes inactive genes because transcription factors are sterically unable to access the DNA. Acetylation by, for example, histone acetyltransferases promotes the loosening of nucleosomes to form euchromatin. And that causes loosely wrapped DNA where transcription factors can access the DNA. And this is all to do with the relative charges of an acetyl group versus a methyl group with the negatively charged DNA and the positively charged histone. And so it's about whether we're causing that to be a stronger attraction or a weaker attraction. Now, in addition, DNA itself can be methylated. So this can occur at something called CPG sites, and that can cause transcriptional silencing. This occurs in promoter regions and is known to regulate gene expression. These are all types of epigenetic modifications in which the gene expression is controlled by things other than the nucleoside sequence. Okay, so now let's talk about pre-mRNA processing. So this is a co-transcriptional process and has three steps: forming the 5' cap, splicing, and a 3' poly A tail. And so this converts the pre-mRNA into our mature mRNA. So first, formation of our 5' cap. So the first nucleotide of newly transcribed pre-mRNA possesses a triphosphate group. This is cleaved off, and a GMP nucleotide is attached in an unusual 5' to 5' linkage. Then the terminal guanine is then methylated on the 7th nitrogen and binds to something called the cap binding complex. Now, this is important because the 5' cap protects the mRNA from 5' to 3' degradation by 5' exoribonucleases, and additionally facilitates the initiation of translation by recruiting the 40S subunit of the ribosome. Now, as we know, sort of in our pre-mRNA, it's a mixture of untranslated regions, our exons, and our introns. And so what we want to get is a continuous open reading frame. And so splicing is an RNA-mediated transesterification reactions, where we have, for each intron that's removed, there are 2 sequential breakages and rejoining of the sugar phosphate backbone. And so this results in excision of introns, fusion of exons, and this also alters the mRNA base because, again, we're removing nucleotides and it can cause a change in frame if it's not a multiple of 3. So pre-mRNA processing is directed by the spliceosomes, and this comprises a large number of proteins called splicing factors, as well as 5 small nuclear RNAs and the pre-mRNA being processed. So these snRNAs have various functions, including identifying splice sites through complementary base pairing, binding to branch sites, and looping the intron into the lariat. Now, there are important recognition sequences within the intron, which are recognized by the spliceosome. And so splice sites are sequences that should be at the boundary between introns and exons, and they are very highly conserved, containing GU at the 5' end and AG at the 3' end. So splice site consensus sequences are also adjacent to these and are also conserved, as well as branch points, which are also highly conserved in the intron sequence, and these are about 40 nucleotides upstream of the 3' AG. Now, why this occurs with RNA, it is a whole long discussion in of itself, but one of the main distinguishing features of RNA is that it has a sugar ribose instead of deoxyribose. And so what you have there is a hydroxyl group on the 2nd carbon of ribose. And so this is actually crucial for the splicing reaction. And so I'm going to go over it briefly, but essentially that hydroxyl group of an adenosine in the branch point directs a nucleophilic attack on the 5' splice site. So that's just a step here. You then have transesterification producing a lariat intron structure that releases the 5' exon. And then you have the 3' OH of the 5' exon, so again here, directs a nucleophilic attack on the 3' splice site, and that results in the fusion of the 5' exon and the 3' exon by a transesterification reaction. And this also releases the lariat intron structure. And so this is how we get our continued open reading frame. So another thing you need to know about is alternative splicing. Now I'm running a little bit short on time, so I'll just say briefly that splice sites can be strengthened or weakened due to enhancing and silencing factors. And this allows us to also do exon skipping. And so what that means is that you can have differential polypeptides produced from the same original gene. And so you can have things like tissue-specific gene expression or differential expression of these factors. Okay. So final step in pre-mRNA processing is the 3' tail. And so it involves two steps, cleavage and polyadenylation. So the major signal for this is this sequence AAT AAA or AAU AAA, which is about 30 bases upstream of the cleavage site. There's also a second signal, which is the downstream sequence element, which is GU rich and located downstream of the cleavage site. So first, a section of pre-mRNA at the 3' end is cleaved. The new 3' end of the 5' cleavage product is polyadenylated by an enzyme called poly-A polymerase, or PAP, which adds between 200 and 250 adenosines in the form of a poly-A tail. And so then this is covered by poly-A binding proteins. So cleavage and polyadenylation are both carried out by the same multi-protein complex, which assembles on specific sequences at the poly-A sites. And so what this does is create a uniform 3' end to all protein-coding mRNAs, and that's really important for the nuclear cytoplasmic export, stability, and translatability of the mRNA. So now we have translation, and so this is the process where we produce a polypeptide from our mature mRNA template. And so this is performed by our ribosomes, our RNA protein complexes, either in the cytoplasm or in the rough endoplasmic reticulum. So we already talked about sort of the generativeness of the genetic code, but I will just briefly touch on tRNAs as something you need to know about. So this is a single RNA strand shaped like a clover leaf because of intrachain base pairing. And so this mediates the conversion between mRNA and protein. So our tRNAs are assembled by amino acyl tRNA synthetases, and these charge the tRNAs with correct amino acids. And it's highly specific, and there's also a proofreading mechanism to ensure its accuracy. So each tRNA molecule carries one amino acid to the ribosome, providing the substrate for the polypeptide synthesis. And it has an anticodon, a sequence of 3 nucleotides as complementary to the mRNA codon. Now, the binding of the anticodon to the codon by complementary base pairing provides the adapter mechanism for conversion of the nucleotide sequence to the amino acid sequence. Now, excluding our 3 stop codons, we have 61 possible tRNAs, but in reality, most organisms have less than 45 tRNAs. And so what that means is that some tRNAs can clearly recognize more than one codon. And so our best explanation for this is something called the wobble hypothesis that says that at the 3rd base, the base pairing rules are less strict, and so it can base pair with more than one complementary base. So ribosomes are our translational machinery, and they're composed of 2 subunits, a large 60S and a smaller 40S, and they're assembled in the nucleolus to form our ATS complex. So the rRNA is a catalytic component of the ribosome, and that's known as the ribozyme. So once the mRNA transcript is exported from the nucleus via nuclear pores and enters the cytoplasm where it enters the cytoplasm where a ribosome associates with it. And so again, as I mentioned before, the 5' gap here is really important for initiating translation as it recruits the 40S ribosomal subunit. And this subunit scans the 5' untranslated region in a 5' to 3' direction for the AUG start codon, and that codes for methionine. Once methionine, the codon for methionine has been identified, the 60S subunit is recruited, and then translation can commence. So next happens elongation, and I always found this a little bit tricky to wrap my head around, but essentially it follows this cycle. So within the 60S subunit, there are 3 sites to accommodate tRNA molecules. So we have our E site, our exit site, our P site, peptidyl site, and our A site. At the beginning of translation, the P site is occupied by the methionine initiator tRNA. And then the A site is lined up with the next codon, ready to accommodate its corresponding tRNA. Once the correct tRNA enters the A site, peptidyl transferase activity of the ribosome catalyzes a peptide bond formation between the amino acids. And so that's between the P and A sites. So you can see here, peptide bond being formed. Then the ribosome shifts one codon along the mRNA transcript, releasing the initiator tRNA through the E site. While the tRNA with the growing polypeptide is then positioned in the P site, then the next tRNA can occupy the now empty A site. And so then this repeats until we arrive at the C terminal. And so what happens is once a stop codon is reached, it's recognized by a releasing factor, ERF1, that promotes the release of the polypeptide and dissociation of the ribosome from the mRNA. So I'll just talk briefly about translation regulation. So first of all, polyribosomes is just to say that multiple ribosomes can translate the same RNA molecule at the same time, and so that allows to produce more proteins. And so that structure is called a polyribosome. Another thing that can happen is an alteration in the phosphorylated state of translation initiation factors, and this can dictate whether mRNA can begin translation. And so essentially this occurs via 4E binding proteins that bind to EIF4E in the cap when unphosphorylated, and this blocks recruitment of the 40S subunit, and so that halts initiation of translation. So this can happen with the insulin signaling cascade, where you have phosphorylation of the 4EBPs, and then you have a negative of a negative, so that prevents binding, and so you have the 40S subunit being recruited. Now, in a similar way, you also have regulatory sequences within the untranslated region that influence mRNA fate. So within that, so for example, within the 5' untranslated region, you have iron response elements. So if iron rises, this binds to iron responsive proteins within a cell. These IRPs bind to iron response elements in the 5' untranslated region. And so if they're unable to bind because they're bound to iron instead, they are unable to inhibit translation of ferritin. And so more ferritin is produced, and so ferritin as a protein sequesters iron, and so that results in lowered iron. And so these examples of insulin and iron are that you need to know about for your exams. Okay. Now we've just about hit 7 p.m. So unfortunately, I'm going to have to stop here. But I can just briefly show the other slides that I would be talking about, just so you can have a flick through yourself later. So organization of the genome is really straightforward, plenty of notes there. And then the last bit that will be talked about is these methods. So that's looking at DNA cloning using restriction enzymes and plasmids, PCR and the various PCR methods that you need to know about, sequencing, including Sanger sequencing and next-gen, and then our nucleic acid methods, so our northern blotting for DNA or northern blotting for RNA, southern blotting for DNA, and fluorescent in situ hybridization, CRISPR-Cas9, and then our antibody-based techniques. You'll have plenty of information on that in the slides, and if there's any confusion, please do shoot me an email. So I'll just skip through to the end. So like I said, my email is here if you have any questions. I'd also highly recommend this resource. So this is a drive I compiled back when I was in first year full of lots of notes. I do believe it's shared around still, but if there's any issue, please use it. And also, I'd really appreciate if you can scan the QR code or go to this link to provide me with some feedback. Great. Thanks very much.